and head of Dendrites Lab at the Institute of Molecular Biology and Biotechnology of the Foundation for Research and Technology, ELAS. Her first degree was in mathematics from the University of Cyprus, and she continued her PhD studies in biomedical engineering from the University of Southern California in Los Angeles. Her research now interest lies in understanding how dendrites contribute to complex brain functions and uses computational modeling approaches, often in conjunction with experiments, to answer this question. She has received numerous of prestigious awards throughout her career, but she is also a member of EMBO and was the first chair of the FENS, Kavli Network of Excellence. Now she will give us a talk with the title Learning with Dendrites in Brains and Machines. Dr. Poirazi, we are ready to hear your presentation.
Thank you very much for the kind introduction. It's a pleasure to be with you today. Indeed, as you said, I'm interested in understanding the brain, and in particular dendrites, which are portions of neurons in the brain. And the talk today is a little bit about the work in the lab towards this goal. So you already had a brief introduction about my career so far. So I was born in Cyprus, and I studied mathematics at the University of Cyprus, and then did a master's and a PhD in biomedical engineering, where I got to know about the brain and how to use my background in mathematics and programming to simulate the brain in a computer. And I stayed in the US until 2000, and then I returned to the Biomedical Sciences Research Center, Alexander Fleming in Athens, where I did a short postdoc in bioinformatics, a completely different new field for me, but still capitalizing on my background in mathematics. And since 2004, I am at IMBB 4th in Crete, where I lead the Dendrites Lab. So as we said already, I'm interested in understanding the brain. And what you see here is a simulation in a computer of the entire human brain. And these little blobs that light up are electrical signals, which is what we use, what neurons use to communicate with one another. And it's really fascinating to understand how this complex system works. And we do that in the lab, starting by focusing at each one of these little blobs, individual neurons. This is a neuron, the main computing unit of the brain. It consists of many parts. The dendrites, which is what we love the most in the lab, and these are the receiving ends of the neuron. That's where signals from other cells arrive. These signals are integrated within the dendrites, travel to the cell body, and then when they reach the axon hillock, if they cross a threshold, so meaning if they're strong enough, there is an action potential generated, an electrical signal, which travels down the axon, and this is then propagated to the following cell. So these electrical signals is the way that our neurons communicate in the brain. And just to give you another visual illustration, this is a simulation again of the Dendrite Gyrus, one of the most important areas in the hippocampus, at the center of learning and memory in the brain. And what you see here are granule cells, the main computing neurons of the Dendrite Gyrus. And here we zoom into one of those cells, and you can see the cell body, the dendrites extending from the cell body and the axon. On the dendrites, you can see these little globs, which are synapses or philobodia. This is the philobodia extending to reach into an axon that passes by. Once it touches these axons, it forms a contact. This is the synapse, and that's how the electrical signals are transmitted from one cell to another. So why do we care about dendrites? First of all, because their properties are altered in many brain diseases, for example, Alzheimer's, Parkinson's, and so on and so forth. And therefore, if we understand how dendrites contribute to normal brain functioning, maybe we can also use them as target for new treatments. The second major role is that they are the main communication site of the neuron. Over 90% of the synapses are formed within dendrites. And the third reason is that they support local computations. And by local computations, I will explain in a while, they transform the incoming signals in nonlinear ways, and thus they can greatly influence processing in the brain. And this is important to understand because we want to figure out how does this system compute information. If we figure it out, we can not only use this information for treating diseases that target these computations, but also to come up with smarter machines. So we do that in the lab using mostly computational methods. We do that because they are faster, cheaper, they require less manual effort, but also because they can allow us to access a level of detail which often is not allowed by experimental techniques. And in this way, we can also synthesize various pieces of experimental data and come up with new discoveries. The types of models that we have in the lab vary a lot in terms of their level of complexity. We have models at the single neuron level, where you can see we represent the detailed morphology of individual cells and capture the various mechanisms, ionic channels, receptors, and so on and so forth that are found in these neurons. And we also have models of small microcircuits. Here you can see one such a model where we connect four pyramidal neurons to one another. When they light up, it means that the cell has generated an action potential, the electrical signal that is used for communication in the brain. We also have more simplified models where we don't capture all this elaborated morphology so that we can describe many different cell types. You can imagine that the more information we put into these models, the higher the computational complexity. So we need big clusters to run our simulations. So there is a trade-off between complexity and simplicity to be able to simulate different phenomena. Now, the interesting part is that these models can also be combined with wet lab setups. Like, for example, in this project, the Eureka project, we are building nanowire arrays on top of which we can culture real biological neurons and connect these nanowire arrays with a computational model so that we can drive activity in the culture, read out the responses, feed it back into the model, and drive it again, forming such a closed loop between the biological tissue and the simulated model in the computer. And this project aims essentially to build a drug testing device where we use the model to simulate Alzheimer's conditions, and we drive the neurons onto the culture while we apply different drugs to come up with better treatments for Alzheimer's. So it's not only, the models are not only used to further our understanding of the brain, but they also can have a wide range of applications, like in this case, in the development of drug screening devices. So I would like now to give you a couple of examples of what we have learned from these models and why using them is important. So the first question that we wanted to address, the most fundamental one, is how do neurons compute? For years and decades, we thought of neurons as point summing devices, meaning that the input comes in the dendrite, but nothing happens there. It's like it passes through. It's like a linear transformation. So whatever comes in, it comes out. And this input is then weighted by a strength of the location of the synapse, summed at the cell body, and produces an output. And this has been a dominant theory for many years. However, now we know that there are a lot of mechanisms in dendrites of biological neurons that could also allow these subunits to compute. And what do we mean by compute? We mean that they can integrate the signals, whatever comes in different ways. For example, one type of computation would be just a linear summation. If the number of inputs that come in increases, so does the output. So the linear summation will be denoted with the line here, the dotted line. But there are other types of computations that can take place. For example, sublinear integration, which means that as the strength of the input increases, the output reaches a plateau and it can no longer grow bigger. So it saturates. Or you can have supralinear integration, which means essentially that initially you start out linearly, and then suddenly the output of the dendrite is much larger than what would be expected if you had a linear integration. So there are at least these three types of computation that could take place in the dendrites. And what we thought or explored so far was just the linear summation. So we used models to answer this question. We built a detailed biophysical model of a pyramidal neuron, the main computing type in the hippocampus. We furnished it with all of the ionic and receptor mechanisms that we've known about and simulated the output of this cell in different conditions. So in this case, we want to figure out how dendrites integrate information. So we placed inputs within the same dendrite versus different dendrites and looked at the response at the cell body. And what we found was that if you place them in the same dendrite, there is this supralinear integration, this boosting computation that takes place within the dendrite. And this is lost when you separate the inputs across two different branches. So this was a prediction at the time, many years back, which we didn't know. We didn't know that dendrites could do this computation, a prediction that came from a model and that later on was verified experimentally in the same neurons in the hippocampus and now also in other cortical neurons. So this is an important contribution of a model to come up with something new and unknown, a new discovery, which then pushes the field forward by generating new research that tests this new prediction. Now, why is it important to understand that dendrites do these supralinear computations as opposed to just summing their units? What is the value for the brain to have such dendrites? So what we went on to show with our models is that if you have many of these dendrites that have these supralinear computations, then an individual neuron is essentially acting like a neural network, where each of the dendrites is doing its own local computations. These are fed to the cell body, and therefore you have one neuron acting as a small network. And we've shown this again in simulations in models by creating such a two-layer network, a mathematical network, and predicting the output of the biophysical neuron. So both the real neuron and the simulated neural network predicted the same output of these cells, confirming in simulations, right, that this is a strong possibility. And we've now taken this approach further to other cell types. This was an excitatory neuron. Now we've done it for inhibitory neurons. And in both cases, we're showing with our models that you have these essentially neural networks residing within an individual neuron. Why is this important? It's important because we know from mathematical theory, but also from our own simulations, that if you have these two-layer neurons, then the storage capacity, namely the ability of these cells to store information, is much higher, up to almost 50 times higher than if you have linear dendrites. So it is beneficial for the brain to have, let's say, smarter neurons because it provides more space for storing information and higher computing capacity. But all this is theoretical, right? It's a mathematical analysis looking at networks versus linear neurons. And this is again a prediction. But we wanted to take this one step closer to what happens in the real brain. So we simulated a biological network where we incorporated neurons with such nonlinear dendrites in these networks. We furnished these neurons with plasticity rules, and we trained them to form memories, much like we do in the brain, right? This is a network. Neurons have these nonlinear dendrites. And after learning, which is too complicated to explain now, but you can read about it in a recent publication. So after learning, what happens is that you have a subpopulation of neurons that captures the memory. So in an entire network, only a few neurons are those that will capture the memory. And these are called engram neurons. And we asked, what is the benefit of having nonlinear dendrites for this particular process of forming a memory engram? What we found, and this is the training that you can see it in a little video, these blobs are synapses. Sorry about that. These blobs are like little synapses. And when they grow stronger, essentially it means that the system has learned. They are plastic. When they're smaller, it means that we are suppressing this signal and the system is kind of forgetting, if you like. So after learning, we have the formation of these engram groups, and we look at their properties. The engram size, so how many neurons store this particular memory, the firing rate, how strong they fire, and their sparsity, how different are they from the rest of the neuronal population. And what you can see is that if you have nonlinear dendrites, then the same circuit can learn a memory using much fewer neurons, much fewer than if you had linear dendrites. These neurons fire at much lower firing rates, and they are sparser, which means they are more easily distinguishable from the rest of the population. So all of these properties are essentially resource savings. They allow the brain to store information using fewer resources. And that's why it's important to figure out what it is that dendrites do so that we can understand how the system takes advantage of these properties. Okay. So the next little example I want to offer is about human dendrites. We know that humans have the ability to perform more complex cognitive functions. So the question is, do their dendrites do something beyond what the mice, let's say, dendrites do, and what could that be? So to answer this question, we collaborated with the lab of Matthew Larcombe in Berlin, and they looked at human neurons from slices from people with epilepsy and cancer. And what they found, oops, sorry, I think I'm out of screen sharing. So what they found was that when they stimulate the dendrites of these neurons, there is a peculiar behavior that was not seen before in other animals. They have these interesting new spikes, which are slower to rise than the typical ones. And they have a very particular feature. As they increase the stimulation, so the current that goes into the neuron, the amplitude of these spikes becomes smaller and smaller and smaller. And this is something that was not seen before, not in mice, at least not so far. And if you want to describe this mathematically, then the transfer function that describes these neurons is what you see here. For a small current, they are not induced at all. For the ideal amount current, you have the maximum amplitude. And then as the current grows bigger, these spikes become smaller and smaller and smaller. And we were wondering what kind of mathematical computation could these interesting new dendritic spikes solve? So we came up with the exclusive OR problem. The exclusive OR problem is a gate. It means that you have two inputs coming in, pathway A and B. If none of them are there, the gate is closed. If the A pathway arrives, the gate opens. If the B pathway arrives, the gate opens. But if both of them arrive, the gate remains closed. And this is, let's say, a complex mathematical problem that requires a network of neurons to solve it. It cannot be solved with an individual point neuron. So we thought maybe these dendrites, which have these transfer functions, are capable of solving this difficult problem. So to do this, we simulated a detailed neuron. We furnished this neuron with this property of having spikes that become smaller and smaller as we increase the current. And we simulated the exclusive OR problem. What this means is that we simulated two input pathways, the pathway X and pathway Y. And we looked at the responses of these dendrites and the neurons in silico. And what we saw that if we activate just one pathway, the response is very strong at the dendrite and at the soma. But if we activate both of them together, the response becomes very small in the dendrite and almost diminishes at the cell body, which indeed verifies that these particular dendritic spikes found in human neurons are capable of solving the XOR problem. So for this discovery, we were featured in Quantum magazine this year as one of the 10 biggest discoveries of the year. We ranked first, in fact, which, okay, is not that a big deal. I mean, we don't have enough time to show you the little video, but if you Google it for Quantum magazine, it can explain why this discovery is very important. And maybe it's one of the reasons that makes us different from mice. Of course, this remains to be shown, right? It's just a hypothesis at the moment. And finally, I can't see the time. So if I'm out of time, please stop me. Otherwise, I have a very small, another couple of slides to share with you. And it's about going into intelligent machines. So understanding how the brain works is important because there is a big now effort to build a less artificial intelligence. So machines that are mimicking how the brain works. And to do that in our lab, we take what we find from our systems or the simulated neurons and try to translate these findings into deep learning systems. So machine learning systems that are trained to discriminate, for example, among different digits. Here you have the 0 to 9 digits. It's a 10-class classification problem, and you want to discriminate images showing the different digits. So what we do is to furnish these artificial systems with dendrites. The only thing that will change is this: each unit that is considered a node or a neuron has now its own dendrites, and these dendrites do not talk to any other neuron. So there is a structured connectivity here. We also use local plasticity rules and other biological features like receptive fields. And we want to figure out, is there any advantage by using dendrites in this system? And what we found, there are too many to list here, I'm just going to highlight one, is that we can get a very good performance using fewer parameters. For example, the state of the art on this problem right now is nearly 100%. So this problem can be solved perfectly, but it requires 1.5 million trainable parameters. So a very big network. However, in this dendritic neuron, in this dendritic model, we can get very close, so 98.1%, not 99 like the state of the art, using 20 times fewer number of parameters. So again, this essentially shows you that what the brain is utilizing as properties is something that is really good for savings, for saving resources, not necessarily for solving the task perfectly, but we can do it by using fewer resources. And that makes sense evolutionary because the brain is under evolutionary pressure, right? We have a limited size of a scalp, we have a limited number of neurons. So the brain had to come up with solutions of solving these problems in the most, let's say, energy efficient way. So we have a little summary here for you. That's all today. I hope I convinced you that dendrites are important. They can contribute to many functions. That modeling is a great tool for studying dendrites because it allows us to investigate questions that experimentally may be impossible and drive new discoveries, and that these models can inspire new developments in other fields like artificial intelligence and medicine. And with that, I'd like to thank you very much for your attention. These are the people in my lab that make all of our work possible and our funders. Thank you for listening.
Thank you very much, Dr. Poirazi. That was super interesting. We see also in the chat that they really like the visualization, and it's something very important and unfamiliar for those that are not in the field. So they like that. And I see that we are a bit out of time, so maybe we can take all the questions from the Q&A, and I would like to remind that we have a Q&A, so please write all your questions there. And maybe we can move on now with the next speaker. Myrto, you take the speaker.
Yes, thank you again, Dr. Poirazi. Unfortunately, we don't have the time for the questions that we gathered, but we will forward them. And with this, I would like to welcome our next speaker, is Dr. Marianna Kapsetaki. She has finished she has received her medical degree from the University of Crete, received it with first honors. Then she had a Master of Science degree in Performing Arts Medicine from University College of London and a PhD in Neuroscience from Imperial College London. In her PhD, she explored aspects of memory in healthy aging and following stroke. During her career path, she has been presented with numerous awards and scholarships, and in parallel with her studies in science, Marianna has distinguished herself internationally, as a classical pianist, having won first prize in 12 international and national competitions and performed over 160 concerts. And with this, I would like to welcome her, and she will talk today with us about eating disorders in musicians.
Thank you very much for the lovely introduction. First of all, can you hear me okay?
It's a bit quiet, if
right.
Maybe, yes.
I'll just go a bit closer. Can you hear me now? I'll just speak a bit louder. Can you hear me now, yeah?
Yes.
Good, good, thank you. So thank you very much for the lovely introduction, and thank you very much for inviting me today to speak to this great symposium. In the past few years, I've conducted research in two fields. It's performing arts medicine and also cognitive neuroscience. So in performing arts medicine, I did research in eating disorders in musicians during my MSc at UCL. And this is the research I'm going to present to you today. And I also, unfortunately, I won't have time to tell you about the research I did during my PhD, where I explored different aspects of memory in healthy aging and following stroke. But if you'd like to learn about these projects, you can follow me on ResearchGate or any of my social media. Some of them have already been published, some are coming out soon, so hope you find them interesting. So let's see. Okay, so we'll start with a little quiz. So what do you think all these celebrities have in common? I don't know if you can answer anything in the chat or anything like that, but I can check the chat, maybe, no. But anyway, I'll reveal the answer. So they're all musicians, and they all have an eating disorder or had an eating disorder during their lifetime, sometime during their life. So here's just a quote from Sir Elton John, a very famous musician, and I'll just let you read it for a little while. It's very interesting what he said about his eating disorder. Okay, so what are eating disorders? So the disturbances of eating or eating-related behavior that are persistent that can lead in changes in how one consumes or absorbs food, and they can lead to significant impairments in one's physical health and also sometimes also in psychosocial functioning. So there are different types of eating disorders. So one type of eating disorder is anorexia nervosa, which is the restriction of energy intake relative to requirements, leading to significantly low body weight. There's bulimia nervosa, which is defined as recurrent episodes of binge eating with compensatory behaviors such as self-induced vomiting. There's binge eating disorder, which is defined as recurrent episodes of binge eating without inappropriate compensatory behaviors like vomiting, like it was in the bulimia nervosa. There's pica, which is quite interesting. It's persistent eating of non-nutritive, non-food substances, such as you can see on this plate, over a period of at least 1 month. Then we have rumination disorder, which is repeated regurgitation of food over, again, at least a period of 1 month. There's avoidant restrictive food intake disorder, which is the avoidance or restriction of food intake leading to failure to meet nutritional needs with at least one of the following, like significant weight loss or significant nutritional deficiency, or dependence on enteral feeding or oral nutritional supplements, or impaired psychosocial functioning. There's also night eating syndrome, which is defined as recurrent episodes of night eating as manifested by eating after awakening from sleep or by excessive food consumption after the evening meal. And there's also like eating disorders non otherwise specified. So eating disorders most commonly occur in females, but there's also high incidence in males. It seems to be underdiagnosed in males. And why should we care about eating disorders? Why are they important? So because they can lead to death. So here's a map of, a map showing you the number of deaths in different countries around the world from 1990 to 2017. So, yes. Okay, also eating disorders are important because they can lead to many complications. So these are just a few complications of anorexia nervosa. So this eating disorder can affect the lungs, the brain, the heart, the skin, the bones, the intestines, like so many different systems in our body. And here are some complications of bulimia nervosa. So because of the vomiting, one can get dehydration or low potassium in the blood, or problems with the teeth, as you can see in the bottom image. Also, other complications.
Excuse me, Marianna, kind reminder, we have 5 minutes left for the presentation so that we can have time for some questions.
Okay. So yeah, here are other complications from other eating disorders. So let me talk to you what I did in my research during the MSc. So I did a systematic literature review in which I screened over 80,000 studies, and I found that there hasn't been much research in eating disorders in musicians. So that's why I conducted a research study, which you can find really online here. So what I did is that I created an online survey which I distributed worldwide, and over 300 musicians participated in this study. And I had different questions, like a question about eating disorders, lifetime prevalence, different risk factors like depression, anxiety, stress, and perfectionism. So what I found is that as a group, most of the musicians in that group were females. The mean age was 31. Most of them were classical musicians. Most of them were instrumentalists. We had an equal amount of students, teachers, professionals, and amateurs, and equal number of soloists and musicians playing within small groups. And most musicians said they spend a great deal of time traveling within one country rather than overseas. So many of them said that they had eaten disorders triggers such as exams, stress, concerts, and most of them said their eating habits didn't affect their career or performance. Most of them said that if they had a higher income, they wouldn't change the diet. Most of them were not addicted to any foods or drinks that feel they may help them as musicians. Most of them were not currently receiving treatment for a medical or psychiatric condition. So regarding the eating disorders lifetime prevalence, I found that 1 in 3 musicians reported having an eating disorder in a lifetime, and these are the most frequent ones reported. And in the eating disorders questionnaire, I found that 1 in 5 musicians report pathological values in the global score, and in the sub-scores, most of them reported having concerns with the shape and weight. So regarding the perfectionism, I found that classical musicians had higher perfectionism than non-classical musicians, and those who reported having an eating disorder in a lifetime, again, had higher levels of perfectionism. And as a group of the musicians, the levels of depression, stress, and anxiety were severe. So in conclusion, as you can see in the middle column, I have all the results of the musicians in my study, and in the right column are results from different studies looking at other populations. So as you can see, eating disorders seem to be quite frequent in musicians, and possible risk factors are the increased perfectionism, depression, anxiety, and stress due to the high demands of their job. Thank you very much for your attention.
Thank you very much, Dr. Kapsetaki, for this interesting talk and for keeping the time. I would like to ask 2 more generic questions. The first one is, of course, the anorexia and the eating disorders are a very common problem in a big part of our population, but you chose to look at it specifically in musicians and artists. Would you like to tell us why?
Yes, because through my experience in the music field, because I'm a classical musician, classical pianist, I noticed that sometimes, especially professional musicians, and especially those who travel a lot, seem to have a lot of stress, depression, anxiety, and especially classical musicians, when they train, like from early, when they're children, they seem to have like very perfectionism habits sort of thing. You're told exactly where to sit, exactly how to have your hands. So all these are risk factors for eating disorders. And I thought, well, let me see if musicians have increased eating disorders. So that's why I looked into the literature and checked if there had been any study, and that's why I did the study.
It is a very interesting point of view, especially because when we think about artists, we never connect them with high levels of stress. So this was a very nice insight. And another question, well, as we said, you received your first degree was in medical, but you chose a somewhat different path, that of the research. Would you care to tell us why you went in a non-classical medical path?
Yes, it's not that like there's a lot of people also doing clinical and research in parallel. So I don't, maybe in Greece it's maybe not so much, but now I'm here in England doing a postdoc. So I found that there are quite a few medics doing research and clinical work, and by doing research, like, it helps you understand better the knowledge from you get from medicine, and it keeps you up to date with everything. And yeah, it's really nice to be doing both research and clinical work.
Perfect. Thank you very much for your talk and for
your
nice answers. Thank you.
So we continue our next talk now with Dr. Barbara Mavroidi. So the title of your presentation is Targeting Central Nervous System Disease with 99M technetium, if I pronounce it correctly. Technetium, okay, and rhenium, agents of high blood-brain barrier penetration potentials. Dr. Mavroidi started her scientific career from the chemistry department of University of Athens in 2008, and in 2016, she completed her PhD study and she continued working as a postdoctoral researcher at the Institute of Biosciences and Applications in the area of the development of new compounds of pharmacological interest. Her main field of research are cancer and neurodegenerative diseases, and among other interests, her research focuses currently on pharmacophore molecules and bioactive natural products, as well as the nanostructure that contain them. Dr. Mavroidi, the stage is yours.
Thank you very much. Good evening. It's a great honor to be here. As you mentioned, my main research interest focuses on the development of novel radioagents of high blood-brain barrier penetration that are mainly focused on the diagnosis and therapy of central nervous system diseases. Let's say a few words about the blood-brain barrier. BBB, as we call it, is formed as a result of the tight arrangement of endothelial cells, packaging of the brain capillary endothelial cells, and its main role is to prevent any entrance of undesired toxic agents and undesired compounds, as well as to keep the microenvironment of the brain as stable as possible. However, owing to the presence of the BBB, small molecular weight drugs with a molecule weight between 400 and 500 Dalton, and approximately 100% of higher molecular weight drugs like monoclonal antibodies, proteins, and genetic agents are failed to pass the BBB. This limitation has contributed to the lack of effective treatments in major central nervous system diseases like Alzheimer, Parkinson, Huntington disease, ALS, brain tumors, and many, many other infections in the brain. It is therefore clear that there is a great and fully justified need for drugs and pharmaceutical agents of high BBB penetration ability. Well, as you mentioned before, my main research interest focuses on Alzheimer's disease and brain cancer. Let's say a few words about the therapeutic approach that they exist right now in these two diseases. As it concerns the Alzheimer's disease, Alzheimer's disease is the most common form of dementia in the developed countries, and I think the 7th leading cause of death, and mainly affects elder people. The histopathological characteristic of this disease is the accumulation of amyloid plaques outside the nerves, and these plaques mainly consist of the beta-amyloid peptide. the current used therapeutic drugs in Alzheimer's disease, they are only symptomatic, and they primarily aimed at the reduction of the disease indications through stimulation of the function of neurotransmission. The last few years, all the research efforts focuses on the inhibition of beta amyloid peptide aggregation. And based on that ability, we have the newly, as you all know, FDA-approved drug for Alzheimer's disease, the Aduhelm, whose use, however, is very controversial, mainly due to its high cost and its ineffectiveness against reversing memory loss and intellectual impairment. Then, as it concerns the therapeutic approaches for brain tumors, brain tumor, the primarily or metastatic, is the third leading cause of death between the age 15 to 30, and is the most common cancer type in pediatrics. And despite the great advance in the field of anti-cancer drugs, its use in neuro-oncology is very restricted, owing to the presence of BBB. And we have a great acquired of drug resistance. And as a result, we don't have an effective anti-cancer drug against brain tumor. Radiotherapy still remains the main therapeutic strategy for brain tumors, in combination with surgical intervention, with low, however, survival rates. The emission tomography, otherwise SPECT, is the most widely used imaging technique, and it is widely available and cheaper in comparison to other imaging techniques like MRI or PET, because it's not depends on the presence of a cyclotron. The use of radio tracers in SPECT tomography, in particular the use of technetium-99m, which are the most widely used radio tracer in SPECT, and this is because it has an ideal lifetime and energy, and it gives us an efficient imaging. And the fact that the technetium-99 and the analog rhenium, that they both have similar optical nuclear properties, similar commercial availability and facile handling, the combination of two, which are considered the hot couple in theranostics, broadens the applications in neuro-oncology. Now, about the aspect for the imaging of central nervous diseases. For Alzheimer's disease, we only have PET radio tracers of F18, whose use is very controversial because they have a very low BBB uptake, and they only work well in well-established clinical Alzheimer's condition. Thus, right now in the market, there exists no imaging agent for the early and accurate and selective diagnosis of Alzheimer's disease. And for the brain tumors, we have the SPECT radio tracers of technetium-99 and two PET radio tracers. Once again, they are not brain specific, they do not cross the blood-brain barrier, and they do not provide accurate image of the brain tumor. Taking all of that together, and after many years of experience in my research team, in the research team that I belong, in 2019, we have discovered families of flat, compact, neutral, lipophilic small and especially low-cost molecules that cross the BBB and they target the CNS pathologies. This is the chemical structure you can see here of these compounds. It's a plain chemical structure. We have a cyclobetadenyl anion. In the place of metal, we use technetium-99 for imaging purposes or rhenium for therapeutic purposes, and they are directly bonded to benzothiazole moiety, benzimidazole moiety, benzoxazole moiety. I don't want to say any more details, chemistry details. I want to emphasize how small and simple molecules they are, and they are very easily to synthesize and with low cost. And we were impressed by their properties. By many, many years of investigation, we discovered that they have a remarkable high blood-brain BBB uptake that is over 9%, which is a very, very, very big precedence. They have a selectivity against amyloid plaques in the brain, the characteristic amyloid plaques of Alzheimer's disease. They are selective to brain tumors, to brain cancer cells, and particularly to glioblastoma cells. And they have many properties, therapeutic properties against Alzheimer and brain tumors. And by making a comparison with the market, with the drug market right now, our compounds are well differentiated in the market of AD and brain cancer, because it is the only 99M technetium radiodiagnostic for SPECT imaging of AD, and is the only BBB penetrating compound in the development of Alzheimer's disease therapy that can act through multiple mechanisms of action. And generally, these compounds have the potential to be used in other neurodegenerative diseases, which are related to protein misfolding, but also to brain tumors and infections. All these results are published in Journal of Medicinal Chemistry, and we rapidly try to protect our intellectual property by registering a patent in, of course, in Greece, in Europe, in USA, in China, etc., etc., etc. As I mentioned before, the research team that I work with is Drs. Maria Spelecanou, who many the other members of the team is Dr. Marina Sagnou, Dr. Minas Papadopoulos, Dr. Ioannis Pyrmetis, and Dr. Antonio Segani. And our work is financially supported by Stavros Niarchos Foundation, General Secretary of Research and Technology, and Ygea Hospital. Thank you very much.
Thank you very much, Dr. Mavroidi. That was a very nice presentation. I have a general question. So you mentioned in your last few slides that these compounds can be also used for other neurodegenerative diseases such as Parkinson's disease. Can you say a bit how this can be used there?
As I mentioned before, we have discovered that our compounds can act as inhibitors of beta-amyloid peptide aggregation. Mm-hmm. Many researchers believe that this peptide aggregation is the main cause of the creation of the amyloid plaques. One of the symptoms in the case of ALS, of Parkinson's disease, is an aggregation of a protein, like amyloid peptide. In the one case is alpha-synuclein, it's the aggregation of a protein of a peptide. Thus, the fact that we have solid indications that our complexes act as inhibitors of peptides aggregation. This can be used in principle and in other type of proteins. This time, we make many research right now to justify this, and the results are very promising that we can also have application in this type of diseases as well.
That's actually very interesting. And may I ask, what type of models do you use for testing that, the inhibition?
Our first screening is with, in the case of Alzheimer's disease, we test it by commercially available amyloid peptide, and we see in vitro how it interacts with this peptide's route of aggregations. Then the next step is the evaluation in cells, in neuronal cells, that we take it from the brain of the mice. And the third step is to evaluate these compounds in transgenic mice, APP mice, that are the typical Alzheimer model. Mm-hmm. So we have, I think, a full screening of the compound's action in vitro and in vivo. As it concerns the brain tumors, we evaluate it in a great variety of brain cancer cells that we have in our cell bank. And we also generate xenografts, tumor-bearing mice, with many types of glioblastoma, neuroblastoma cells. And we see in vivo the results of the administration of these compounds.
Very interesting. Thank you very much for answering the question. We have also one in Q&A, as I see now. Thank you, Dr. Mavroidi. How do these molecules cross the brain endothelial monolayer? Do they activate an innate response, changes in PMN levels, or the endothelial monolayer, adhesion, molecule abregulation, etc.?
We try to find the mechanism that they pass the BBB. It's something that we have to see about. I don't have the answer. I only have the effect, the results.
Okay, thank you very much. I don't see more questions in Q&A.
I would like to ask someone, since we still have a bit of time. I would like a first question concerning these molecules. What is your pool? Do you do it by screening a library? Are they products that they come from natural products, or they're only chemical synthesized?
One of our favorite compound that we have many years experience in that is the benzothiazole, the family of benzothiazole. And we do many optimizations in this chemical structure. And we do the medicinal chemistry part. We change something in the structure and we test it. But we screen a great variety of natural products like curcumin, like many extracts of green tea, and we have many promising results. But the best results right now was with this kind of molecules.
Okay, thank you very much. And with this, we would like to thank you and go on to our next and last and important speaker for this session. Perfect. Thank you very much.
Thank you. So I hope that you can hear me well.
Yes, we can hear you well. Thank you.
Right. So as Myrto said before, I'm Marta Vardaki. I'm currently a postdoctoral fellow at the University of Ioannina. I'm based at the medical school. I'm part of the medical physics lab, and I will talk to you about Raman spectroscopy. That's sort of the second imaging technique that will be mentioned in this session, I think. I'm from a small island in the North Asian Sea. This is Limnos. When I finished with the school, I moved over to Patras, where I got a bachelor and a master's degree. Then I moved over to the UK for a biophysics PhD, followed by a couple of postdocs in Imperial College London, and then the University of British Columbia over in Vancouver, Canada. And then just a couple of years ago, I relocated back to Greece under a fellowship from Kiki. And this is where I'm currently based at the University of Ioannina. So I am actually trying to solve medical problems using Raman spectroscopy for the past 10 years. And so I will just very briefly try to explain to you what Raman spectroscopy is. So it's a type of light-matter interaction. So light can get reflected or absorbed or transmitted or scattered through matter. And Raman effect is a very special type of scattering. So Raman was initially discovered by Raman back in 1930s when he got a Nobel Prize for that. He actually started thinking about it when he was observing the color of the sea, and this is he was thinking why it is blue, and that's not because it reflects the color of the sky as most of us think. It's actually because the sea tends to scatter more efficiently the blue wavelengths of the sunlight. And just to remind you at this point that the white light is made up of many different colors. So when I say wavelength, I essentially mean the blue color. And the normal case is that when we shine the light beam onto a surface, that this is being reflected back in the same color without changing color or properties. But what Raman observed was that a very, very tiny amount of this light, which is maybe about 1 out of 10 million photons, is being reflected back in a slightly different color. And this is the Raman light. And the most impressive thing about that is that when the light hits different material, the colors that are scattered back are different. So this will be different for a bone or for a healthy cell or for a population of malignant cells. And this effect comes down to the different way that the molecules vibrate inside the samples. So there are different molecular vibration, and in the same way, the light interacts in a different way with them. So on the left-hand side, we can see the Raman spectrum. This is the typical output of our measurements. Now, every single peak corresponds to a specific molecular vibration, and then the height of the peak is, generally speaking, relevant to the concentration of the molecules inside the sample. And now, because the Raman spectrum is very characteristic to each material or cell or sample, call it whatever you want, it's like something like a molecular fingerprint. So that allows us to work the other way around. When we have a sample of unknown origin, we can actually take it to Raman spectrometer, record the spectrum, and then go and identify the unknown sample. And this is my whole work. This is what it's actually based on. So we are trying to collect this very, very weak Raman light. And the most common way to do that is by using microscopes, Raman microscopes, as we call them. People are using them to look into biofluids. to identify biomarkers which may be related potentially with diseases, and then also to look at the distributions of those biomarkers over the sample area. Also, we're going to use them to look into bone and cartilage diseases, where we have studied the changes in the mineral part of the bone or in the organic content of bone and cartilage, which is mainly made of collagen. And another more recent application, which we demonstrated recently, is to really be able to investigate the interfaces between those two tissues, which are of major interest. And here we looked at vertebral end plate, which has been now shown to be implicated in this structure, one of the main causes of back pain. Now, another very exciting application from microscopy is being able to look at cell cultures. And so what we are picking up when we are recording Raman spectra are the spectral signatures of the all those macromolecules which coexist in the same like sampling volume of the cells. And those can be lipids or proteins or nucleic acids, and then we can either target specific biomarkers there, which of course need to be of a high enough concentration, or we can look at general changes of those macromolecules. And of course, using our Raman data, which can be thousands and thousands of spectra, especially when we are doing mappings, we can then go back and reconstruct them into images where each one of them can show a Raman peak of interest. For example, if we want to see the spatial distribution of a protein or a specific lipid over the sample area. And in that way, we can really analyze the cellular and sometimes the subcellular features as well. Now, another application from microscopy, which is quite recent as well, is monitoring the cell differentiation status. And that means if we have a cell that is actually undergoing a differentiation towards another cell, we can really look at the changes that it's going through. So, for example, if in that case we have an embryonic cell which is being differentiated towards maturing beta cells, and these are cells with an immense potential for diabetes cell therapy. So the challenge now with the differentiation protocols is that they can take so much time and they can cost so much money. And another thing is that at any random stage, the cell can deviate from the expected truth. And because of money and time, we should be able to we should be able to realize that this has happened. So we really need to ensure the consistency of the cells at each stage, and also for the end product to be safe. And Raman spectroscopy is a tool which can really help us here, because every specific stage of the differentiation protocol usually provides a very distinct Raman spectrum. Now, if we really want to take to make a good use of cell therapies and take this to a manufacturing level, so to actually deliver those cells to thousands of people, we need to scale up, so move from cells to cell culture bags to bioreactors, and all of those without while maintaining the consistency of the cell material. And for that reason, the Raman tool is something that also needs to evolve. There are actually Raman tools in the market, which are able to do that. So, for example, you can see here on the right-hand side the Raman probe, which is able to monitor specific markers in a cell culture environment every few seconds. However, you can also see that this is a tool which does not take advantage of the non-invasive nature of Raman spectroscopy, because the Raman probe is completely immersed into the cell culture. So what we've been trying to do for the past few years is really develop a non-invasive approach of Raman spectroscopy. So while we, in the conventional form of it, we are only able to collect information, Raman light, very superficially from the surface of the sample, we have exploited the way that the photons propagate in the sample. And then we have decided to collect them from the point where they emerge, and that essentially means to take the collection path more far away from the excitation and collect the Raman photons, which are produced deeper inside the sample, and so essentially collect information from deeper inside in the sample, and at the same time avoiding the signal, the Raman signal from the container. Either it is the wall of the bioreactor or the plastic container of a flask. We managed to do that in a cell culture of Gyrus cells last year, where we were able to record the Raman signal from the cells without opening the bag and without breaching the sterility of it. Now, this approach, we also employed it in another application, and we actually used it to look into the quality of the blood, of the stored blood, which is destined for transfusion. So when a donor goes to visit a blood bank, his or her whole blood is being broken down into individual blood components. Those components have a different shelf life. They are kept under different conditions and for a different period of time. The red blood cells specifically, they can be maintained for up to 7 weeks in the fridge. However, it is a current problem for the blood banks that it's completely impossible to test every single bag without breaching the sterility of it, because they cannot be open. They are sealed until they are delivered to the patient who needs them. And so this is an issue because we now know that once the red blood cells are stored in the plastic bags, in the transfusion bags, there's a series of biochemical changes that take place. They all include the oxidative stress as well, and they're under the umbrella of storage lithium. So these changes lead the quality of the red blood cells to degrade. And on the top of that, there's also the donor variation, which means that the blood for every donor is not aging in the same way. For some donors, this is happening faster. For other ones, it's happening slower. But unfortunately, there's no way for us to know which blood is aging faster or slower. We cannot predict that. And so this is where the deep Raman spectroscopy comes in, because with the non-invasive measurements, we were able to actually track down the oxygenation of hemoglobin, which has been shown to increase with storage time. This has been also correlated with morphology of the red blood cells, which is a marker for the storage lithium. We have picked up changes in the metabolism of the red blood cells, where the lactate increases with time as a product of glycolysis, whereas the glucose is dropping. And also it was, we were very happy to see that for different donors that was different, as it should be, so that the donor variability was able to be reflected. And then it's important to say that these measurements can be done completely non-invasively, and they can also be correlated with biochemical data, which can happen in parallel. For that, we have actually used some very advanced heteromodel correlation analysis. This is also a very big part of my field.
Dr. Vardaki, I'm sorry for interrupting. Just to remind that we have 3 minutes to wrap up and have some time for questions.
Yeah, that's my last slide. So just before wrapping up, I just wanted to show you a couple of applications of Raman spectroscopy, which you can really find very close to the clinics. On the right-hand side, the top right, you can see that the doctor is actually scanning with a laser the leg of a patient. And in order to assess the quality of the bone underneath the skin of the leg, and so in that case, the patient was diagnosed with osteogenesis imperfecta. And in the other case, there's a video here with a surgeon shining a laser onto the brain of a patient so that he will be able, after recording a Raman spectrum, to assess the type of abnormality and the surgical margins better before I take the biopsy. And with that, I would like to thank all of our funders, the collaborators over the past 10 years, all of you for your attention, Eleni from the Real Science and Lina from the Greek Women in STEM. All of the organizers, you guys are doing an amazing job. Please reach out to me if you want to discuss anything, and thank you all.
Thank you very, very much for this talk. I would like to ask a generic question before we finish. How difficult was it to pursue the career that you have, coming from a different field from your basic educational, like when you finished your degree? You combine 2 very different fields.
I think it's been quite challenging to go from a sort of biological medical background, because I'm a pharmacist by training, moving over to a PhD in physics or biophysics. That has been the hardest part for me. But as I always say, if you have the right people next to you, and if you really want it, there's nothing that can stop you, right?
Thank you very much. And with this, we would really like to thank all of you for these amazing talks, all you exceptional ladies. A reminder that we follow with our panel discussion, and we really look forward to see you there. And for more, please join us for networking session to meet with our speakers and meet all of us. Thank you very much.
Thank you.
Καλησπέρα. Είμαι στη σωστή θέση, Ακούτε καλά?
Yes, yes, we can hear you.
Okay.
We will, I think we're starting in 2 minutes.
3 minutes, okay, I'll be
Yes.
here.
Okay, I think we can start. Hello, everyone. Welcome to the panel discussions for Biosciences. Today we have the pleasure to introduce to you 3 wonderful women scientists who are thriving in biology. And we're ready to discuss, ask questions, and enjoy. We invite everyone to submit your questions throughout the entire session in the Q&A section, and we will address them to the panelists at the end. So it's time to introduce ourselves. Me and Katerina, we're going to be the chairs of this session. A few words about me. I'm Maria Vlastara. I'm a chemist. I'm currently a final year PhD in nuclear medicine based in the Netherlands. And my role in Greek Women in STEM is mainly a science writer and editor. Along with me, I have Katerina.
Hello, everyone. I'm Katerina. I'm a dentist, and I'm currently in London while doing my master in health science in Sweden. In Greek Women in STEM, as Maria said, I'm a science writer as well, an editor, while running our social media, specifically our Instagram account. So if anybody reads through us through Instagram, probably has talked with me. So let's continue to meet our 3 brilliant scientists here. We have Dr. Kapsetaki. Welcome. She's a biologist and postdoctoral researcher in cancer evolution at Arizona State University and pianist with over 160 recitals across Europe. That's impressive. Dr. Papadopoulou, a biologist as well. Welcome, Dr. Papadopoulou. Professor in plant biotechnology at the University of Thessaly, member of many committees for conferences and symposiums. And we have Dr. Stamatiou, who's a biologist as well, a postdoctoral researcher in respiratory medicine and pharmacology, teaching of many courses at the University of Thessaly, and a mentor in Greek Women in STEM. Welcome, Dr. Stamatiou.
Welcome, everyone. So one of the first things that we would like to ask you is that all of you come from the same background. So all of you are biologists, although you took totally different turns in your career, specializing in very much different fields. So Dr. Kapsetaki, when do you think was the pivot moment that you can share with us briefly when you specialized in your current field and deviated from biology?
Yeah. So first of all, thank you for having me in this session. Maybe I'll talk a little bit about when I did my PhD at Oxford, it was focused on the evolution of multicellularity. So that's all about how single cells cooperate to form a group. And like the pivotal moment was just seeing how cells cooperate. And then I was interested in how they, like this group kind of breaks apart into single cells. And that's very similar to cancer. So by looking at one phenomenon going one way, I was very interested then in my next career path, which is like, I'm now looking at cancer across species. So yeah, one field switched to the other. It's quite interesting, yeah.
So it's kind of the flow of all your steps. Very interesting. So I would really like to know if also, Dr. Papadopoulou, did you also have a similar way of reaching to your current field?
I'm the oldest here. I think so. I have a long history, and I'm not very sure I can find the pivot moment when I started, but choosing plants was a serious decision, I think, many years ago, because even today with what we have heard, all the talks and what I've been experiencing all these years in academia and research, working with plants, it's not the first priority for most people, because we don't consider them very interesting organisms. Well, I thought differently from the beginning. And for me, working as a plant scientist was my main target and decision, because I think they are really, really important and interesting organisms. So I've been working with plants for all these years from many different aspects, and I have been working with plant biology as it integrates with all other disciplines, actually.
And on the other hand, I guess, Dr. Stamatiou, you're not working with plants. No,
not
and another field. A biologist again, but not plants.
For me, the moment that I realized that I want to do research in physiology was during university, when I was first introduced with cell physiology and signaling pathways. And at that moment, I knew that that's something I want to do. And when I started my PhD, I found really exciting to work with inflammation, both chronic and acute. And the one led to the other, and the respiratory system led to cardiovascular, and back at the respiratory that I'm working right now. Signaling pathways were really interesting to look in. So it's still going. I don't know where it will go, but it's still going.
That's very nice. I think this will be very inspiring also for all the people who are watching us now in the earlier stages of their careers. And another question that we were thinking when we were looking at your CVs is that, can you tell us maybe a field that no one can expect, but can be combined with biology? I don't know, who wants to go first? Dr. Kapsetaki, you're smiling. Maybe you have a nice answer to share?
Yeah, recently I was thinking about, like, I mean, throughout my career, I haven't really combined music and biology. My sister has kind of a bit, but I'm thinking recently of how can sound inform us about diseases across the tree of life. Like we know, for example, that male whales communicate via sound, and like the ships that travel kind of disrupt their communication, but it can also affect their stress levels. And we know that songbirds, they communicate via singing. And so anyway, there are a few species that we are very well studied in terms of sound, but I think it would be interesting to look at how music is combined with biology much broader.
Yeah, I think that, I don't think that many people would have thought music, to be honest. So I think that's very interesting indeed. Yes. I don't know if the rest of you, do you have any other Any other answer?
If I can, yeah, can I take the?
Of course,
yeah.
take the lead, yes.
Well, for me, one moment of revelation really was attending a conference back in 2013 in synthetic biology, where one of the key speakers in the conference was a fashion designer. And I've never thought about this until that moment, that biology can mix up so closely and bring new products to this field. For me, it's a field that I know absolutely nothing, of course, but apparently the designers and the fashion needs new materials. Our world and our societies, they need new sustainable ways of producing textiles, and biology can bring these new products through the biological processes by using procedures that they are used by microorganisms to produce new materials, like I don't know, new textiles are produced by algae, like weed. They are produced by fungi, mycelia, by microorganisms, and I think this is really fascinating. A completely new link of biology with the real world, producing also contributing to sustainability, which is one of our main targets, I think, nowadays in biology. So I'm using this example when I teach biotechnology to my students, and I tell them that biotechnology can take you anywhere that you want, even in GNTM, you know, in a career that is completely out of your you know, of our this little world that we know as the cells, disease, and plants and animals. It can really bring you to any type of career that you can imagine.
Yes, yeah, I think I 100% agree on that. Dr. Stamatiou, you want to add something?
I think that one of the most fascinating things about working with cells is seeing different stuff. And photography is a kind of art that cells are really amazing. After all these years of working with such things, I cannot but be excited when I'm looking in the microscope and see a cell that it is stained with some fluoroform, some chromoform, and has a really beautiful cytoskeleton, or you can see a protein that sparkles all around the cell, and it's really art. I think that that's really beautiful. I have been using photographs of our experiments as pictures in collaged in the walls of on the walls of the lab, and it looks great.
Very nice, very interesting answers, really. Both the art and science combined, and also the biology is, well, it can be on trends, right?
And yeah, biology, as it says, is the study of life, right? So is there any cool thing discovered in your field that you would like to share with us, like one cool thing? Dr. Kapsetaki, you're smiling again.
Yeah, I'm thinking about a research project I've been working on recently on looking at cancer across species. And as I was looking through the database, I was noticing the different cancer prevalence of each species. So there's high and lows, but it's a whole mix of species. And just by looking at the names, because most of the scientific names are Greek, I mean, I was looking at the types of animals, and I noticed that lots of the carnivores had lots of very high cancer prevalence. So that reminded me of like what we know of humans, that like red meat is often linked with colorectal cancer, whereas like vegetables and fruit are often on the healthy side of the diet. So a cool finding is that we see essentially the similar things in across the tree of life, that carnivores seem to have lots more cancer than the herbivores. That's something that amazed me, that's similar things that we see in humans across so many species.
Right, there must be many of those. That's an interesting one to share.
One species, yeah, lions, panthers, reptiles, lots of the snakes, yeah, versus like, and lots of birds, they have a lot lower cancer rates. Lots of birds that live in the tropics, frogs, fish. So it's like, if we think of ecology, there's a lot of different trophic levels, and we kind of know that from basic biology that there's this process called biomagnification, that toxins accumulate in the tissues of animals at higher concentrations, at higher trophic levels. So it kind of makes sense that there's probably more toxins in the tissues of the carnivores, so that might be related to the higher cancer rates that we see.
And Dr. Papadopoulou, do you have a cool thing discovered?
Yeah, I think this is a difficult question.
Yeah.
Because science is full of cool things, and every day we learn something new. If I may speak of something bigger that we are experiencing the last years, is the knowledge that we have about symbiotic and beneficial relationships, in starting from the microbiome in the gut and the microbiome with other relationships, also with plants, and realizing that success in biology and what is really matters is this good relationships, and that it is, I mean, biology has focused on disease, has focused on in the past on pathogenesis, on pathogens, on how to fight the bad guys, but apparently, well, it's clearer, at least more and more, that maybe the secret is in finding out what is happening with the good relationships, and maybe the balance is there, and maybe we have to look more into official and symbiotic relationships, and maybe by nurturing this, we can lead to a better balanced biological systems. And this, I think, becomes apparent more and more nowadays. And I think this is fascinating. It's a completely change of mindset of how to deal with dysbiosis in general. And I'm talking about a general theme in biology now, because we see that in humans, we see that in animals, we see that in plants, and that we have to look more into beneficial relationships.
Thank you.
I think this, and I won't say the secrets of plants and their uniqueness.
Thank you. Another question, which I think that I also saw it on the chat, asked by a few of our participants, is regarding the communication and the outreach aspect of science. Dr. Kapsetaki, before when you mentioned art and specifically music, because we know that you're a pianist as well and also a scientist. I was wondering if you have seen similar patterns or any similarities that there are in music, but also in the laboratory.
Yeah.
Are there any?
I can tell you that, I mean, certainly I've noticed that one field helps the other. Like for me, it's communicate. I mean, playing on the piano on a large stage is essentially communicating with people. And in the same way, giving presentations is communication with people. And I see that it's helped me when I give science talks. It's helped me be more confident with talking, just being exposed to the large audiences. But essentially, music and science share lots of things, like in terms of critical thinking, like when I go and practice a piece of music, there's always like some structure in the piece that I have to analyze. There's like an introduction, like a middle area and a conclusion. And we see the similar kind of structure and different voices and phrases. Again, when we write a publication, we have to think of the different paragraphs, and each paragraph has to have like a point, message has to be clear. Just general points that each field has helped me quite a lot. And of course, both need like, you have to be devoted for some time in each field, and you need to study it quite well to be.
And practice a lot.
Yeah.
Fascinating.
And I guess you have to bring your own mind into this, your own soul, right? This is also important because each one of us has a specific style, I think, in accessing science as well.
Yes, as well, like
music for sure.
Yeah, when we write a publication, we always bring our own style, like in the writing, right? So,
yeah.
Okay, so now that you said about the writing, Dr. Stamatiou, you are, I think you have a big experience with students and mentoring. You're also a mentor in our group for quite some time, but I'm guessing that since you are also teaching at the university for many years, I'm guessing you have a lot of students approaching you as a mentor. So have you been ever asked any, what is the more common question that you get asked by all of these students?
I think that it is if someone can combine research and life, especially in Greece. If someone can do research in Greece and live from research, make it a profession. That's a question that often is asked by students. Can I do research and stay in Greece, or do I have to go abroad? There are so many students that are fascinated by research experiments, and they want to do research, but they stop just because they believe that you don't get paid being a researcher, or you can't have a normal social or family life when you have to work for countless hours in a lab. And especially in Greece during the recession, it's really hard because they know that there are very few fundings for research, very few research fundings, and everyone thinks that it is one way, just to go abroad and work there. And I practically don't know what to answer, because I know that it is hard. I know that sometimes in order to be a researcher, you have to do something else as well in order to gain money and live, just pay your bills. And it needs to be, for someone to be very committed on the dreams they have, on the focus they have in research, and someone must really love to do things like that in order to be successful, and just to cope with all the difficulties that may come in the way. But if you love something, you will find a way to do it.
Yes, I think yes. I think in every profession you need to devote yourself and love it, of course.
Let's proceed with another question that interests us. So you're three unique women in research, right? And as we see and we assume, you have managed to achieve your ambitions, or at least a great part of them. So
I cannot hear you very well. I'm sorry again.
Sorry, now can you hear me?
Better, yes, thank you.
Okay, so I was saying that we assume that you have managed to achieve your ambitions, or at least a great part of them. So could you outline some people that helped you the most in your career? Like, was there someone that tried to unlock the potential into you? Dr. Papadopoulou?
Yes, well, there are always people around that will help. even teachers in elementary school can push you, and help you. You need to be a little bit lucky sometimes. You need to also to be open to this. And yes, there were always, I can think of people since really my youngest years, but I think the big switch has happened when I decided to do my first postdoc, because at that moment I decided, it was my conscious decision to join this lab, which was led by a young female PI. Not very famous, It was in a very good institute, the research institute. So the scientific community was there, but I choose to be close to a person, to a personality, And this was, I think, very important, because she was a very good scientist. First of all, very organized, very good in her science, but she was also a good mentor. And I've heard this word again and again. Very important to find somebody that can help, that can be honest and supportive. And for me, this was the person. Her name is Anne Osborne. She's a senior scientist now. I feel a bit, it's not fair, because she was not, of course, the first one to help, but sometimes we need these people that will make, that will help us realize our potential, and and Anne Osborne was one of them. So if we are here to help young people, because as you are, to decide, I think this is an advice, if I can call it advice, that I can that I always give to my people in the lab as well, that choose to be near to right people, not the famous and the big names, because this can help you only on the scientific, maybe get one big paper, but you need to be able to stand on your own feet and decide that you can be the leader in the next big paper. And this can be done only if you believe in yourself, and this can be done only if you are in the right environment to do so.
Right, that's very encouraging for everyone being in academia.
Yeah.
Yeah, so we had another question thinking with Maria. So we were wondering, have you ever felt that you need to be unreasonably overproductive to prove yourself constantly in academia? Did you ever feel that way? Dr. Stamatiou?
Most of the time. Well, when you work in a Greek university, especially as a postdoc, and as a PhD candidate before that, you have to do things because it is understaffed. So you have to be involved in other things than your research program or your thesis. Sometimes you have to do lab work, you have to do teaching, help with supervising, teach techniques to other researchers. At the same time, find the economics, find where the antibody costs less in order to order it, and you have to do a lot of stuff. That in some labs abroad, it is unthinkable. A researcher most of the labs has to do just his work or her work, but you know, doing other stuff at the same time, it really makes you realize that you can do more stuff than you thought you would, and it teaches things. You can know your limits and go beyond your limits, and every day you can know that you can gain more possibilities. Since you have more things you can do, you learn new things, even things you wouldn't dream about, such as zooming in. We all had to do something like that in our careers. It's interesting. It's hard when it's done, but it's interesting. You can achieve new things every day.
Dr. Kapsetaki, would you like to answer to the same question?
Yeah, I'd like to add something that has happened, like now with the pandemic. Many people are working remotely, so there's a big difference in hours. For example, I'm in Greece at the moment, but most of the people I teach or I work with are in Arizona. So there's like a 10-hour difference, which means I have to work like over hours. So usually my meetings start after 6 until sometimes 1 o'clock at night. So again, we have to, I mean, I'm testing my limits, but like it's a difficult to like this work-life balance. It's quite difficult, especially during the pandemic. I'm working remotely, yeah.
That was actually one of the questions that we just had on the chat. Any advice on work-life balance? I think this is a really big topic.
Just go by the day.
Just go by.
Just go by the day. You can plan anything. Whenever you plan something, it's meant to be wrong. You can plan anything. Really, every program I have ever made collapsed in my face. Just go by the day.
And Dr. Kapsetaki, for example, you are also combining music and the lab. So have you found the perfect balance, or are you struggling? Or
again, it's like organizing things day by day. So every morning or
day by day.
Yeah, and luckily I have people around me, like my family, that helps me organize some concerts, like on the organizing part. But again, in the same day, I have to organize when to practice the piano and read, or it's every day. I've kind of got used to it over the years, but yeah, it's a struggle.
Yeah, but it's important. It's important that you you set your limits, and you have you know when you have to work, when you have to take a break, and when you have to take time for your family. It's very important. Listen, I have been through periods that I was working around the clock, and it's fine if you do it for a short period of time, if you have specific deadlines and targets, but it should not be your reality, because it's important that we do other things. I think that Dr. Kapsetaki is lucky to have this other thing that is also important, because this other thing is not, again, the same you know, everyday life in the lab, or administrative work, or teaching, and it's also something, it's art, okay, and art elevates you and brings you to a different level. But we all have to find, even if we are really lab rats, as I used to be when I was young, we have to find other things that can give us balance, and it's important. And try to have to build a different mindset, I think, now that we are young, and make time for your other things. If I'm allowed to say something, the person that I mentioned before, Anne Osborne, you know, she's a very good scientist, as I said, a very top scientist now, but she's also a poet. She's writing poetry, and she takes months out of her lab and of her science career to go on writing poetry. I think this also brings you, down to earth and makes you realize and gives you strength to go on, because being a scientist, as Dr. Stamatiou said, it's very stressful and frustrating most of the times. Okay, we love science and we love the research and the lab, but it's tough to do science. So we have to find ways to let the steam out and bring and energize ourselves.
And even combine them. Why not? I've also encountered people who were painting, they were painters and also doing a PhD, and they combined it, and now they're scientific illustrators. I think, maybe you can combine everything.
So I guess we have a recipe here. So go day by day. You both agree on that in every level, academic level. So all of you come from different academic levels. And have you noticed any differences in female presence in your field at each level that you've been? Dr. Stamatiou?
There are less women doing research than men. I think that when it comes to making a family and having kids, it's tougher for a woman. And the try to combine things is even tougher. And sometimes you have to cut on sleep or on living just to make through the day. And I think that that leads to many women to not to be involved in science and even quit science, even though they love it, even though they have good, a fine future in front of them, they choose to quit because they think that you have to choose between kids and research. It's not the same. I believe that research is another kid you have. And me personally, if I didn't have a strong support system, my family, my husband, my kids, my mother, my mother-in-law, all of them helped me do what I dream to do. And it's tough.
Yes, thank you.
Thank you. It is tough to combine everything.
Yes, and women disappear from science. It's true.
As you go.
If you go in the biology department or in our department, the biochemistry and biotechnology department, we have more women than more female than men, and slowly they disappear from the labs, yes, it's tougher for women to climb, especially to climb up to the different positions, especially in academia. It's true, you have to make a lot of compromises, and this is one of the things that we have to fight for still, especially in Greece, because we don't have the society is not ready to support women as we should. We expect our mothers to bring up our children, which is not what it should be done. I will dare to say that our partners are not trained yet to support us as equally. Well, at least you are younger, maybe you are experiencing different things, but for our generation, it has been very difficult. We had to, I mean, I'm also a first generation graduate, a degree holder, because my parents, they didn't finish university. For younger generation, this is easier now. So you had to convince first your parents that what you are doing is right. and then your partner to support you. So this is something we need not to fight on our own. We need to make the societal changes in the society. We have to fight for this still, but this doesn't mean that we cannot make it.
I think that maybe this is common in other professions as well. I'm guessing.
Yes.
Yeah. So maybe it's good to say that. And I don't know if have you ever thought if do you think that the university itself could have a certain support system for parents to help them advance in careers? We've seen that in big companies, also abroad, they have like a different system for support.
Abroad, you mean?
Yes. And maybe we could see that in the universities tomorrow?
I don't know.
Far from that.
Yes, we are far from that. I mean, one example, my PhD student, she wanted to have a baby and she could not take leave, maternal leave. We had to fight with IKI, with the university, with everything, as if you are doing it, when you are doing a PhD, you cannot have you cannot be a mother at the same time. This doesn't exist in the system, and it's not only the Greek system. Don't take me wrong. Even in the EU, if you have a Marie Curie fellowship, maternal leave is not in their plans. You cannot apply for this type of leave, So it's strange. It's strange. We are still a little bit back with all these balances and women presence in science.
Yeah, balance is really hard to be kept. Though, how do you counter these difficulties in your field? I assume it's because you really love science and you really love to be in labs and you love what you do and you love waking up and go to the lab. So there must be a way that you can counter this roughness of science. Is there one, Dr. Kapsetaki?
You mean how we manage with all these difficulties? I mean, it goes back to, I mean, we do whatever we love. I mean, in any field, if you love something, you do it no matter the struggles. It might have been better if the government supported women in a different way, but I think anyhow, we would still be in science. Well, I mean, the essential thing is to talk about it and be open about it and try to find solutions. That's the first step. And then if we could talk with like key politicians maybe and try to take it to the next level. I suppose, I'm not sure, like if in Greece, there could be like societies between different departments and university representatives that talk to politicians about these issues. I think there needs to be some more kind of links or people to express the problems within the university to try and change things with the government. I think there's a bit of a gap there. It sounds like everything's perfect, but there's certainly lots of small things and bigger things that could be changed.
Yeah, sure, that's a nice idea. And Dr. Stamatiu?
I personally took my kids with me in the lab.
Oh, you did?
Yeah.
I think I've also heard that many times from supervisors.
The first time I took my daughter to the lab, she was about 3 years old, no, 2 years old. And when we were just about to reach the university, she said, "Oh, we have to go back." And I said, "Why? We haven't taken any food to feed the cells. We have to go back." Yeah. And from that day on, she knows that cells drink juices. Strawberry juices, because the medium is red. So I just take my kids to the lab. I have a support system around me, and I'm trying to do stuff. I think that I'm really lucky that in my lab, most of the faculty, of the academic members of the lab are women. and they pretty much know what a woman has to go through in order to do research. And yeah, we're one big happy family. We know each other kids because everyone has brought their kids to the work, and we always have crayons in the lab so that kids can draw stuff at the moment that mom is doing an experiment, and they can be at school. So yeah, practically we're combining it by introducing children at a very young age in science. That's the way we try to see it. That we're not just doing it, we're introducing them to science. It's good though.
But you're still
It doesn't always work.
Yeah, I know. I know, that's why I said that you have to have a support system. And unfortunately, the university at this moment, it's not going to give you a support system. So I think that
it's not bad.
Yeah.
Yeah, it's not bad to know that you need a support system and plan for it. Yeah. And then you can do it. You know, this is important.
Yeah.
And support system is not just our mothers. Huh?
Yeah.
It can be friends. It can be people that maybe we can exchange time with them. There are ways to do it, and of course we need to support each other. As Dr. Stamatiu said, if you are with other women that they know what it means to have a, for example, I don't know what type of problem, then it can help. We have to support each other.
That's true. That's why we all are here today, to support each other and know that we have to.
And well, there is a solution for everything apparently. So that's really nice. Moving on to some questions from the chat and from our participants. So I have a question for you, Dr. Papadopoulou. Knowing how demanding and time-consuming it is to be a principal investigator and a lead group of researchers, how do you manage to combine that along with full-time professorship? Is it difficult?
Maybe not so good. I don't know. Anyway, for me, the hard part is the administration. Okay, the administrative work, which is a lot as you go higher in academia, and then this is a lot of extra work that you don't do science, you don't mentor people, you don't educate people, you don't read, and you just sign papers, and you do all this extra work that needs to be done so that your lab is running and your students are, as I say, well fed and supported. And this is a lot of work. This is for me the hardest part. Not having lab members and mentoring, because this is actually my fuel. The energy comes from this. From the people in the lab, because usually they are brilliant people, really talented people. Because younger people, they have so many more perspectives in their lives. They are very well educated. You read a lot and in many different fields, and it's an inspiration to be with young people in the lab. I like teaching, luckily, but I'm telling you that I'm not managing very well because the administrative work is horrible. And apparently not only in Greece. That's the main complaint of PIs, I think. I don't know if I have answered. Yeah.
I think yes, it's your fuel. That's what I'm taking from this.
Yes. Yes, but it's true. It's true. The people in the lab it's for me it's such a pleasure to have that talented people in the lab, honestly. I wouldn't be here without these people, of course. You don't manage on your own. You need your team.
You definitely needed to proceed. So, Dr. Kapsetaki, you have achieved so much so early. You're a pianist with so many recitals across Europe. Do you have a message for all the girls in our audience that they're feeling shy and think they cannot make it? Is there an advice to just try? What do you have in mind?
I mean, every person is different. Like throughout my life, I just read and read and read and read, and that has led me to where I am. I think and support from the family and everything, but maybe I've missed on other things in life. I don't know, going out to the cinema or going to bars every night. I can't give I would say for each person to find their individual goals or what fulfills them in life. That's the most important. Like, I don't think I can give like a general piece of advice for every single person, because each person has their own interests. They want to spend their time in a different the day devoted into different things. Like if they want to become a pianist, they would need a different kind of schedule every day. If they want to become a scientist, they might have to try different things. Yeah, there's not a one way recipe, I would say, to a success, or and success is you can't define success. It's so many different ways. I mean, people are successful in cooking, and my parents or anybody, they're excellent cooks, or each person is, I can't define success. So if they're happy, that's success for them. Like if they're happy with what they're doing every day.
And what about ambition? Because I'm guessing that you also need that as another fuel. Yeah,
it depends on your goals again. I mean, if you want to become a pianist, you would need to study in a certain way. If you want to become a cancer biologist, you might need to go into those research areas. Or again, it's not one recipe. Like somebody could study astrobiology, and I know some people that have studied astrobiology and now they work on cancer. Or it's so these days with the information like floating so broadly everywhere, like somebody can spark a new idea by combining completely different fields, and that might be the new Einstein in this era. It's exciting in a way. Yeah, it's not one recipe as we maybe think in some traditional villages here in Crete. They might think you need to do this and this and that and become a farmer, and that's your goal in life. And that's set from a very young age. But today, I think it's exciting that we have a bit more freedom as women. If we compare with what many years ago, we had the freedom of explore so many different things and travel. So I think we're privileged in many ways.
Yeah.
So those are the positive news compared to the other generations. Yes. We're moving forward.
Yes.
Yeah. So let me ask you, have you received any specific treatment from male colleagues and mentors during your academic journey? Like, was there something specific that you would like to pinpoint?
Is that for everyone? Or?
Sorry?
Is it for everyone? Who was that?
Yes, it was a question, I think, from the Q&A. Yes. So, Dr. Stamatiou, do you want to answer? No.
What kind?
If you have experienced something that you would like to pinpoint.
Good or
maybe,
bad?
Yeah. Sure, well, whatever.
Well, a bad one. I was trying to get to a master's program after I did my PhD, because there were some new techniques that when I finished biology at the university, they weren't used. And I thought that I needed to know those techniques in order to take my research to a new level. And at the interview, I was told that I was too old. I was 38 years old at the time. I'm now 41. Okay. I was too old to do research. And I was, okay. So if you think that someone is too old to do research, then I don't want to do research with you. I don't think that it had to do with being a woman. I think that it had to do just with the perspective some people have about research. Unfortunately, there are some people that think that research is my research, and I have to have staff to do my research, to answer my questions. It's not my research. Just like Dr. Papadopoulou earlier said, it's the team's research. And we work as a team. And it's not my data. It's not my questions. It's the team's questions. So it doesn't matter how old you are, if you're a woman or if you're a man or whatever, what's your nationality, whatever. It doesn't have to do with it. You're just another brain, and another brain can bring different aspects in a team. So I think that even when looking at bad behaviors, you can find ways to come out stronger. And see that probably it went for the best, because I think that it would be a waste of time after all. Yeah,
we really like that perspective. We admire that.
And you can learn things from everyone, from younger. I get things from my students every day. Every day. I have to mention this. Lina Doku, our own from Greek Women in STEM, really opened a new way for me, because she pushed me to get out of my comfort zone and do things that I would never imagine doing. And she's much younger than me. We have different experiences, but you can learn things and get to another level by everyone. You can't just say that he's younger or she's a woman. I don't have to hear about her, what he or she has to tell me. Yep, you can gain things from just interacting with people. And that's why teams work.
That's the reason why we're doing this symposium also, to interact and network and inspire each other.
That's exactly the point, yeah.
There is a question in the chat that I would like to answer very shortly and briefly.
Yes.
Because I think, I don't see it now, but there was a question if you should come back and return to Greece
from abroad.
Yes, yes, of course, yes. Because Greece will not change if you are not, if we don't change it. Who is going to change the situation? And the Greek universities, and research in the Greek universities is in a very good level. I mean, you heard all these talks this morning from from Dr. Poirazi and the other, I mean, this afternoon, sorry, you know, because I've only heard the Biosciences session, but I'm sure that in other disciplines it's the same. I mean, we have people in the lab from all over the world. And why not Greeks? Of course, Greeks. I mean, we have from China, from Brazil, from Belgium, from Sweden, from England. If they can do research here, you can do research as well. So I advocate people to go abroad, because there you can see how other things work in a different environment. But go there, learn, and bring it back, and update and enrich and diverse the Greek environment as well. And we can make a difference. You can make a difference. The young scientists, the young women scientists, the scientists that they are abroad. Yes. do your research in the Greek universities and the Greek research centers.
I think a lot of people want to have this chance.
Yes.
So
well, I'm not very
we'll
sure.
see.
I mean, it's not very easy to find Greek postdocs nowadays.
That's very interesting.
Yes, I know this is another story.
I guess that's the quote of the day.
Thank you for the invitation. Thank you.
Oh, you're most welcome. So yeah, all good things come to an end. So the Biosciences session. We invite everyone to give us feedback or ask questions or anything in Greek Women STEM social media, either Facebook or Instagram, LinkedIn, Twitter. We're almost everywhere. So a huge thank you to all of you, the audience, our brilliant speakers, my partner in crime, Maria, that held successfully the whole conversation today. So the next session gonna be mathematics and physics, 6:45. So see you in the next session.
See you. Thank you.
Bye.
Thank you for the invitation.
Bye-bye.
And congratulations. You should do it
Thank
again.
you. We will.
We will.
Yes.
Bye-bye.
Bye-bye.